Now, today's lecture of related to cell structure and functions, we will study about what are biomolecules, and this is the lecture 1 for the chapter biomolecules. So, starting with the introduction, we know that as an organism consists of various organ systems. These organ systems are in turn made up of various organs, and these organs are composed of tissues which in turn are composed of cells. So we study that the cell itself contains various organelles, and these organelles are the we can say if we study the structural hierarchy in the molecular organizations of the cells. So we will see that at the level 4, there will be the cell and its organelles. Now these cells as a whole is the smallest structural and the functional unit we know of the life. But the cell is further composed of many small molecules, which we will get to know are known as biomolecules. So at the level 4, if I'll see the cell and its organelles, then for example, if I consider that a cell is made up of, for example, here it is shown a plant cell. So the plant cell consists of a cell wall, it consists of a plasma membrane, it has a nucleus, which itself contains supramolecular complexes like chromosome, which are the condensed form of chromatin as we study. So at level 3, we see that there are many there are many compositions like cell wall, plasma membrane, or the chromosomes which are present. These are known as the supramolecular complexes. And these supramolecular complexes are further made up of many macromolecules. For example, the chromosome, as we know, is the condensed form of chromatin. And chromatin is nothing but it's a DNA. So at level 2, we have macromolecules. For example, if we look at the chromosome, it is made up of DNA. If I look at the plasma membrane, it is made up of lipids and it contains proteins. If I talk about the cell wall, it is made up of cellulose. So then at level 2, we have macromolecules like DNA, protein, cellulose, etc. And these DNA, proteins, or cellulose are made up of further composed of many monomeric units that occurs at the level 1. So for example, DNA, that we will study later when we get to know more about the biomolecules, that DNA, which is a nucleic acid, is made up of smaller units, which are known as nucleotides. So DNA is a polymeric chain of nucleotides. Similarly, proteins are composed of many different amino acids. So a chain of amino acids leads to the formation of a protein. And cellulose is made up of many different sugar molecules. So carbohydrates, for example, there are many sugar molecules like glucose we know. So these form the monomeric units at the level 1. So this diagram basically represents the structural hierarchy in the molecular organization of the cells. So in this plant cell, the nucleus is an organelle containing several types of supramolecular complexes, for example, chromosomes. And the chromosome consists of macromolecules of DNA and many different proteins. And each type of macromolecule is further made up of simple subunits. For example, as I mentioned, that DNA is made up of nucleotides, and the full form of DNA is deoxyribonucleotides, for example. So this is just an example to show what is the composition and what are the further smaller units which are present inside the cell. So there is we know that all living organisms are composed of cells or products of cells. Now there is a wide diversity in living organisms in our biosphere. We know that we have different flora, we have different fauna. There is a wide, huge diversity of living organisms in our biosphere. So the question is that are all living beings, we know that all living beings are made up of cells. But the next question is, are all living organisms made up of the same chemicals, that is the elements or the compounds? Is the chemical composition of all the organisms is almost same, or is it different? So to analyze this, so to have the answer to this question, we need to analyze the elements or the compounds that makes up a living organism. So the answer to this is all living organisms are made up of the same elements and compounds. A cell is composed of variety of molecules, for example, carbon, hydrogen, oxygen, which perform various functions. For example, if we consider about the glucose, so glucose is nothing but C6H12O6. So it is made up of carbon, hydrogen, and oxygen. And the combinations of these carbon, hydrogen, and oxygen leads to the generation of different chemical compounds. Or for example, as I mentioned, glucose, or these carbon, hydrogen, oxygen may also combine to form amino acids. So these different units perform the different functions. And other than these basic elements, some metals and non-metals are also present as cellular materials. Now, if we perform an analysis of a plant tissue, animal tissue, or a microbial paste, carbon, hydrogen, oxygen, and several other elements will be obtained, suggesting that irrespective of the type of living organism, the basic composition of all the living organisms is almost same. That is, it is mainly composed of carbon, hydrogen, or oxygen, and may include some other different metals or non-metals. The same analysis made of, if we make the same analysis on a non-living material, for example, if I take a piece of Earth's crust and I try to find out a list of the chemicals which are present inside the Earth's crust. So we have found that, like, not we, but the scientists have found that the Earth's crust, the chemicals which are present in the crust of the Earth are almost similar to the chemicals which are present inside the human bodies. So the chemical analysis suggests that most of the elements which are present in the crust of the Earth are similar to the ones which are present inside the human body. All these materials combine in different ways in order to form various biomolecules. As I mentioned, that these carbon, hydrogen, oxygen may combine to form sugar molecules. These carbon, hydrogen, oxygen molecules may combine to form an amino acid that are the units of the protein. So the combination of these carbon, hydrogen, and oxygen may also lead to the formation of various lipid molecules. So these materials then combine in different ways in order to form the various biomolecules which are found in the living cells of the organisms. These molecules, one specific thing about biomolecules is that these molecules are itself not living. Proteins are not living, but they perform various living functions. So these molecules are not living, but they perform various living functions. Thus, biomolecules, what is the definition of biomolecules now? So, thus, biomolecules are the organic substances, for example, carbohydrates, proteins, lipids, etc., that play a major role in the structure and function of all the living organisms. I repeat it again. Biomolecules are the organic substances, that is, they are non-living materials, non-living molecules. Example, carbohydrates, proteins, lipids, etc., that play a major role in the structure and function of the living organisms. For example, we know that glucose, being a sugar molecule, helps in gaining the energy. While similarly, if we talk about the lipids, we know that lipids and proteins are the part of the cell membrane. They are the part of plasma membrane. So they play a major role in the structure and function of the living organisms. Besides these, water is also an important and most abundant chemical compound which is present in the body of the living organisms. We know that 70% of our body is composed of water. So water is also important, and it is the most abundant chemical compound which is present inside the body of a living organism. So how do we get to know that what is the chemical composition of the living organisms? So for that, we can perform the chemical composition analysis. And to analyze the chemical composition of the biomolecule, what we can simply do is, for example, if I want to analyze the chemical composition of let's say a leaf, or let's say for any plant tissue or for animal tissue. So what we will do is, we will place that tissue sample in the trichloroacetic acid. And upon reaction, it will then form a thick slurry. And this slurry is then passed simply through the cheese cloth. And the one which easily gets passed through the cheese cloth will form the acid soluble pool, or which is the filtrate, while the other one is the which remains above the cheese cloth only is known as the retentant, or also known as the acid insoluble pool. Like the one, the items of the tissue, the materials of the tissue that gets dissolved inside the trichloroacetic acid will be passed on through the cheesecloth material and will come into the acid soluble pool as a filtrate, while the other material will remain in the insoluble form, and this will be known as the acid insoluble pool. The acid soluble pool contains chemicals with smaller molecular masses. For example, the chemicals with a molecular mass of 18 to 800 daltons approximately, and these are known as the micromolecules or biomicromolecules. They include amino acids, sugars, nucleotides, etc. While the acid insoluble fraction contains molecules of larger molecular weight, of heavy molecular weights that have a range above 10,000 daltons, and these are known as, since they are greater in size, these are known as macromolecules or biomacromolecules. These include polysaccharides, proteins, nucleic acids. So basically, the micromolecules, for example, amino acids, combine to form the macromolecules, for example, protein. So micromolecules are in turn the kind of, we can say, they are the, for some micromolecules, we can say that these are the units of the macromolecules. For example, as I mentioned, amino acids are the units for the protein. Sugars, for example, are the unit for polysaccharides. Nucleotides are the unit for nucleic acids, example DNA and RNA. Now, beside chemical composition analysis, we can also perform the analysis of inorganic elements which are present within the living organism. So inorganic elements and compounds are also present in the living organism, and they can be known with the help of ASH analysis technique. So what we can do is, we will take a small amount of a living tissue, for example, let's say again a leaf or a liver tissue, and we will just weigh it without drying it. So the weight will be known as the wet weight. And then we will dry it and weigh it again. So due to the process of drying, all the water will get evaporated. So now whatever the weight we will get is the dry weight. Now when the tissue is fully burnt, the carbon compounds are oxidized to form the gas like CO2. Water vapor is removed and the remnant is called the ash. This ash contains many inorganic materials, many inorganic elements like calcium, magnesium, etc. So in the acid soluble fraction, inorganic compounds like sulfates, phosphates are also present. So elemental analysis gives the composition of living tissue mainly in the form of oxygen, carbon, hydrogen and nitrogen. Also sulfate, phosphates are present in the acid insoluble pool. So this is about the inorganic element analysis if we want to perform in the living tissue. So what we will do is, we will simply take the dry weight. Firstly we will take the wet weight, we will dry it, then we will take the dry weight. We will burn the tissue. Due to the process of burning, all the water will get evaporated. All the carbon, that is the organic material, will get oxidized to form the gases like CO2, and therefore it will get removed, and then we will perform the simple elemental analysis to check what are the different elements which are present in the living tissue. So what we found is, there are many chemical elements, for example, calcium and magnesium, that are present, while in acid soluble fraction, inorganic compounds like sulfates and phosphates are present. So this is a comparison of the elements which are present in the non-living matter, example earth crust, and the living matter, example human body. So we can see that the composition is similar, but the proportion may vary, like percentage weight of the elements in earth crust and human body may vary. And oxygen is the most abundant material element which is present inside the human body as well as in the earth crust. So this is very important that oxygen is the most abundant material, most abundant element which is present inside the human body. Also, it is all the abundant element which is present in the earth crust. While there are other elements like hydrogen, carbon, nitrogen, sulfur, sodium, calcium, magnesium, or silicon, which are present inside the earth crust, as well as like except silicon, all other elements are present inside the human body. So silicon is not present inside the human body, or it is present in negligible amounts. Similarly, there are various other, besides these elements, there are various ions which are present. So this is the list of inorganic constituents which are present inside the living tissue. So elements ions like sodium, potassium, calcium, magnesium, water molecule, compounds like sodium chloride, calcium carbonate, phosphate and sulfates are present inside the living tissue. They form a inorganic constituents of living tissue. So there are various functional groups that are present in the biomolecules. We get to know that biomolecules are mainly made up of the carbon, hydrogen, and oxygen. And these molecules gets arranged in different functional groups. So there are various functional groups that may be present inside the biomolecules. For example, methyl, ethyl, phenyl, and carbonyl, which is an aldehyde. So aldehyde may be present in some of the biomolecules. Similarly, amido group, guanidino, and imidazole may be present. That these we will get to know, like in various amino acids, imidazole ring is present, or similarly in various amino acids, amino group or carboxyl group are present. So the other functional groups that are present in biomolecules may include carbonyl, carboxyl group, hydroxyl group, ether or ester. They may also have anhydride as a group. They have sulfhydryl, disulfide, thioester as a group. They may have phosphoryl, phosphoanhydride, or mixed anhydride as a functional group moiety that may be present in the biomolecules. So these are the functional groups which I mentioned that may be present in the biomolecules, and they may impart the functional property to that particular biomolecule in which they are present. Now, beside biomolecules, what are metabolites? So there is another term which is a metabolite. So the internal volume of the cell, which we know is made up of cytoplasm, which composed of cytoplasm, is an aqueous solution, the cytosol, and a variety of suspended particles with specific functions. So basically, cytoplasm is made up of aqueous solution, which is known as cytosol, and various functional particles are suspended in this aqueous solution, in the cytosol, forming the cytoplasm. So the cytosol is a highly concentrated solution containing enzymes and the RNA molecules that encode them. The components, for example, amino acids and nucleotides from which these macromolecules are assembled, is made up of hundreds of small organic molecules which are called metabolites. So these metabolites are basically the small organic molecules that further leads to the formation, that may be the components of the macromolecules that get assembled and therefore perform a specific function. Metabolic pathways can lead to a more complex structure from a simpler structure. For example, acetic acid may become cholesterol, or it may lead to a simpler structure from a complex structure. For example, glucose becomes lactic acid in our skeletal muscles. So there may be the formation pathway, there may be a pathway in which the degradation may happen. So during the metabolic pathways, sometimes it may happen that a complex structure may be getting converted into a simpler structure, or on the other hand, it may also happen that a simple structure is forming a more complex structure during the metabolism. So the former case is known as the biosynthetic pathway or the anabolic pathways, where a more complex structure is formed from a simple structure, while the later, in which the complex structure forms the simpler structure. This pathway constitutes the degradation pathway and it is known as the catabolic pathways. And anabolic pathway plus catabolic pathway as a whole is known as the metabolic pathways, or the pathways that leads to the degradation or the formation of new molecules, either the complex molecules or simpler molecules from some complex entity, is known as the metabolic pathways. And the molecules that take part in these metabolic pathways, the small organic molecules that take part in these metabolic pathways are known as the metabolites. So metabolites may be of two types, primary metabolites and secondary metabolites. So the basic difference between these is primary metabolites, as the name suggests, is the primary thing which is needed by the cell. So they perform the physiological functions of the body. So they are like very important, and they while secondary metabolites are the derivatives of the primary metabolites. Primary metabolites play the direct role in growth and development of the organism, while the secondary metabolites take part in the ecological functions. Primary metabolites are same in all the plant species, while secondary metabolites are unique to each plant species. Primary metabolites are produced in large amount and extraction is comparatively easy, while for the secondary metabolites, they are made up of, they are produced in small amount by the plant cells and the extraction is comparatively tough as compared to the primary metabolites. This is the list of some primary metabolites and secondary metabolites. So some of the primary metabolites may include enzymes. We know that enzymes have a very important function in the survival and in the growth and development. So enzymes like protease, lipase, peroxidase, they are the examples of primary metabolites. Similarly, amino acids like proline, leucine, they are examples of primary metabolites. Organic acids like acetic acid, lactic acids are examples of primary metabolites. Vitamins like A, B, C are examples of primary metabolites. While if I talk about the secondary metabolites, the various pigments which are produced by the plant cells or various toxins which are produced by the plant cells or the polymeric substances, for example, we know the gum. Gum is a very common polymeric substance which we know, or rubber, we know that some plants exudates rubber in the form of rubber, secondary metabolite in the form of rubber, so which is a polymeric substance and is an example of secondary metabolites. So secondary metabolites may include following, for example, pigments like carotenoids and anthocyanins. It may include alkaloids, for example, morphine or codeine. It may include essential oils, for example, lemongrass oil, rose oil. It may include toxins, for example, abrin and tricine. It can include lactants, for example, concanvaline A. It may include example like drugs, for example, vinblastine and curcumin, or polymeric substances like rubber, gums, and cellulose, all comes under the category of secondary metabolites. As these metabolites are not the primary thing which is required by the cells or the plant tissues or the plants for the development or for the survival. So they play a role in ecological functions, but not in the physiological functions of the body. So this is the list of the primary metabolites and secondary metabolites. Next is different types of biomolecules that may be present. For example, sugars that are example of carbohydrates. For example, as I was continuously mentioning, glucose. Similarly, ribose is also an example of biomolecules. Amino acids that may, for example, lead to the formation of proteins. So these amino acids may include, for example, may include glycine, serine, or alanine. Fats and lipids are also an example of biomolecules. For example, fatty acids like palmitic acid, glycerol, or triglycerides. Phospholipids like lecithin, which forms the plasma membrane in some cells, or cholesterol are examples of biomolecules. So starting one by one with each of these, the first one is amino acids. Amino acids are the organic compounds that contain amino group and an acidic group as substituents on the same carbon. As we can see in this figure, that in amino acids, for example, in glycine, alanine, or serine, we can see that if I take, for example, alanine, we can see that we have an amino group and an acidic group like COOH which are attached on the same carbon. So we can see in all the amino acids, we have an amino group and an acidic group which are attached on the same carbon. So which is this carbon is known as the alpha carbon. So hence they are called alpha amino acids, and they are nothing but they are substituted methanes. Now there are 4 substituent groups occupying the 4 valency positions, and they are, so we can see in the diagram that if I take, for example, methane is CH4. So if I start substituting one hydrogen with each group, we can see that, for example, in alanine, we can see that one hydrogen is substituted by COOH group. The other is substituted by any other R group. For example, in case of alanine, it is CH3, while the third carbon is substituted by amine group or NH2 group. So based on the R group which is present, there are 20 different types of amino acids which are present inside the human body. So there are 4 substituent groups occupying the 4 valency positions, and these are hydrogen carboxyl group, amino group, and a variable group designated as R group. And based on the nature of R group, there are many amino acids which are present in the living organisms. However, those which occur in proteins are only of 20 types. So these 20 types of amino acids combine in different orders to form a different types of proteins. The R group in these proteinaceous amino acids could be hydrogen, for example, in case of glycine, if we will see. So it is the simplest amino acid in which the R group is nothing but a simple hydrogen moiety. And it may be substituted by a methyl group, for example, in case of alanine, or it may be substituted by a hydroxy methyl group, for example, in case of serine. The chemical and physical properties of the amino acids are essentially of the amino group, carboxyl group, and the R functional group, which is attached to the alpha carbon atom. Now, based on the number of amino acids and carboxyl groups, they may be further classified into acidic or basic type. For example, there are acidic amino acids like glutamic acid or aspartic acid. There are basic amino acids, for example, lysine, histidine, and arginine, and neutral amino acids, for example, valine amino acids. Similarly, the R group is an aromatic chain, is an aromatic group. So these will impart the aromaticity to the amino acid, and these are amino acids will be known as the aromatic amino acids, for example, tyrosine, phenylalanine, and tryptophan. Now, since a acidic group is present, a basic group is present in an amino acid, so a particular property of amino acid is the ionizable nature of NH2 and COOH group. So in solutions of different pH, the structure of amino acid changes depending on the ionization of NH2 and COOH group. So this is the structure, this is the table, or this is the diagrammatic representation of different structure of different amino acids. Also, we can see that there are various amino acids, for example, alanine, valine, isoleucine, leucine, methionine, phenylalanine, tyrosine, tryptophan, in which the hydrophobic side chain is present. Hydrophobic side chain is present. While there are certain in which aromatic rings are present, for example, we can see in case of phenylalanine, tyrosine, and tryptophan. So based on different properties of the side chain groups which are present in different amino acids, we can classify amino acids into different categories. Now, I was talking about the ionization property of the ionizable nature of NH2 and COOH group. So because of that, it may acquire either a positive charge or it may acquire a negative charge in solutions of different pH, and there might be a pH where both the groups are in the ionizable form and the net charge on the amino acid is 0, or you can say that the net charge is 0 on the amino acids. So this form of amino acid in which both the groups are ionized and the net charge is 0. This is known as the Zwitterion form of the amino acid. And the pH at which the amino acid occurs in the Zwitterion form is known as the isoelectric point. That is Pi of the amino acids. I'm repeating it again. Now, since in an amino acid, there are two groups which are present, for example, NH2 and COOH, and both of these have the tendency to get ionized, and depending on the solution, pH of the solution in which the amino acid is present, they may ionize into different forms. And there might be a certain pH at which both the groups are ionized such that the net charge on the molecule or the amino acid is 0. This form of amino acid in which the net charge is 0 is known as the Zwitterionic form of the amino acid. So in this figure, the B is called the Zwitterionic form of the amino acid in which the net charge on the amino acid is 0. The next type of biomolecules are the lipids. Lipids are generally water insoluble and they could be simply fatty acids. A fatty acid has a carboxyl group attached to an R group. The R group could be a methyl, for example, CH3, or it can be a ethyl, C2H5, or higher number of CH2 groups. It can be 1 carbon to 19 carbons. For example, palmitic acid, we all know, is a 16 carbon long lipid fatty acid, including carboxyl carbon. And arachidonic acid has 20 carbon atoms, including the carboxyl carbon. Fatty acids could be saturated. Saturated, we know, without the double bond. Unsaturated means with one or more than one carbon-carbon double bond. So fatty acids may be saturated or they may be unsaturated. Another simple lipid is glycerol, which is trihydroxypropane. Many lipids have both glycerol and fatty acids. So here the fatty acids are found esterified with the glycerols. So simply lipids are fatty, we can say that in most of the cases, lipids are fatty acids that are found esterified with the glycerols. They can be then monoglyceride, diglyceride, or triglyceride. And these are also called fats and oils based on the melting points. Some lipids have phosphorus and a phosphorylated organic compound found in them. These are called phospholipids. We know that phospholipids are an essential component of the plasma membrane. So they are found in the plasma membrane. And I mentioned one of the example, lecithin. Lecithin is one such example. And some tissues, specifically the neural tissues, have lipids with more complex structures. So neural tissues have structures which are known as myelin sheath that are made up of lipids. So this is about the lipids, and we know, for example, there are various fats and oils like palmitic acid, as I mentioned, is a 16 carbon, including the carbonyl group. Glycerol, or it may be a triglyceride, in which we have like triglycerides in which R1, R2, and R3 are the fatty acids. Phospholipid, example, lecithin, that may be present in some cells, or cholesterol that may be present inside the plasma membrane. The next is a nucleotide. So living organisms have a number of carbon compounds in which the heterocyclic rings can be found. Some of these are nitrogen bases, for example, adenine, guanine, cytosine, uracil, and thymine. So adenine, guanine, cytosine, uracil, and thymine are the nitrogen bases that are present in the nucleotides. Now, in nucleotides, when when we attach sugar moiety to these nitrogen bases, then this is called as the nucleoside. So if I attach sugar moiety to, for example, deoxyribose or ribose to adenine, guanine, cytosine, uracil, and thymine, then this is known as the nucleoside. And if a phosphate group is then attached to a nucleoside, then the complete unit is known as the nucleotide. So basically a nucleotide is a combination of firstly nitrogen base, then sugar, and then phosphate. If I talk only about the nitrogen base and sugar, then it is called nucleoside. And if I add a phosphate group to a nucleoside, then it is called as a nucleotide. For example, adenosine Guanosine, hymidine, uridine, and cytidine are nucleosides. And adenylic acid, hymidilic acid, guanylic acid, uridylic acid, cytidilic acids are nucleotides. And nucleic acids like DNA and RNA consist of nucleotides only. And DNA and RNA, we know they function as a genetic material. So these are the examples. For example, nitrogen bases like adenine, which is a purine, and uracil, which is a pyrimidine. So the easy way to remember purine is GAPPU. GAPPU is guanine and adenine are the part of purine. So remaining uracil, thymine, and cytosine are the part of the pyrimidine. So guanine and adenine are the part of purine. And to remember this, we can easily remember GAPPU. GAPPU means guanine and adenine are purine. So these are the nitrogen bases. As I mentioned, when we add a sugar moiety to these nitrogen bases, they form the nucleosides. For example, adenosine, uridine are the examples of nucleosides. And if I add a phosphate group to these nucleosides, they will form a nucleotide. For example, if I add a phosphate group to this adenosine molecule, it will form an adenylic acid, resulting in the nucleotide. Now, this was about the biomolecules. There is a term called biomacromolecules. So, biomacromolecules I mentioned in this introduction slide. So basically the ones which are present in the acid insoluble fractions are the macromolecules. So there is one feature which are common to all the compounds which are present in the acid soluble pool. That feature was that they have a weight ranging from 18 to 800 daltons approximately. While in case of acid insoluble pool fraction, we have four types of organic compounds, mainly example, proteins, nucleic acids, polysaccharides, and lipids. These classes of the compounds, with the exception of lipids, have molecular weights in the range of 10,000 daltons and above. For this very reason that these biomolecules, that is the chemical compounds which are found in living organisms, are of two types. The one is the micromolecule, one is the macromolecule. And for which have weight less than 1000 dalton are referred to as micromolecules or simply biomolecules, while those which are found in the acid insoluble pool or acid insoluble fraction are called as macromolecules or biomacromolecules. So lipids, here I talk that lipid is found in the acid insoluble pool irrespective of the fact that its weight is below 10,000 daltons. So why lipids? Therefore, it is said that lipids are not strictly macromolecules. Lipids are indeed small molecular weight compounds and represent not only as such but also arranged into the structures like cell membrane and other membranes. So whenever we grind a tissue, we are disrupting the cell structure. And cell membrane and other membranes get broken into different pieces if I grind the tissue. And upon grinding, since the lipids are not water soluble, they will form the vesicles. And these vesicles, irrespective of the fact that lipids are of low molecular weight, will retain over the cheesecloth and will come into the acid insoluble fraction. Therefore, these membrane fragments in the form of vesicles get separated along with the acid insoluble pool, and hence they come under the macromolecular fraction. The acid soluble pool therefore represents only the cytoplasmic composition. The macromolecules from cytoplasm and organelles become the acid insoluble fraction. Together they entirely represent the chemical composition of living organisms or tissues. So basically, if I talk about lipids, proteins, polysaccharides, they come under the acid insoluble fraction. Now, the average composition of the cell includes the water, protein, carbohydrates, lipids, nucleic acids, and ions, of which, as I mentioned, water is almost the 70% to 90% of the total cellular mass. So, till now, we get to know that the major biomolecules which are present inside the living organisms are lipids, nucleic acids, carbohydrates, and proteins. These biomolecules are made up of further smaller units of micromolecules. For example, nucleic acid is made up of nucleotide, carbohydrates are made up of smaller sugar monosaccharides, proteins are made up of amino acids. Similarly, lipids are made up of glycerols and smaller units. So biomolecules can be carbohydrates, proteins, nucleic acids, and lipids. Carbohydrates have the monomers, example monosaccharides, which are joined by glycosidic bond. Examples include starch and glucose. Elements include carbon, hydrogen, and oxygen. And what is the function of carbohydrate? They are the energy source, structural component, and they act as the reserve food material inside the cell. For the proteins, monomers are the amino acids which are joined by the peptide bond. Examples may include the insulin and the collagen. They composed of different elements, for example, carbon, hydrogen, oxygen, nitrogen, and sulfur. Example function may include that most of the proteins, like most of the enzymes are proteins. So therefore, they have enzymatic role. They have the function in the structure and movement. They also help, like they are also a part of defense hormones. Nucleic acids are made up of monomers, which are nucleotides that are joined by a phosphodiester bond. Example include DNA and RNA. They are composed of various nucleic acids, for example, carbon, hydrogen, oxygen, nitrogen, and phosphorus. And this stores the genetic information within the within itself, and they get during the process of reproduction, they get transmitted from one generation to the next generation. The next is lipids. Lipids are made up of monomer units like fatty acids and glycerols that are joined by the ester bonds. And examples include the fats, oils, and waxes. And the elemental composition of lipids is mainly composed of carbon, hydrogen, and oxygen. Again, lipids are good source of energy. They act as an insulator and provides the insulation. They are important or essential components of the membrane, and there are certain hormones that are present in the form of lipids. So in short, if I have to summarize today's session, I can say that biomolecules can be like all the composition or the material within the cell can be divided into two types, organic and inorganic. Organic materials may include macromolecules and micromolecules. For example, macromolecules may include polysaccharides, polypeptides, and polynucleotides. Micromolecules may include lipids and secondary metabolites. These polysaccharides are further made up of simple sugar. Polypeptides are made up of amino acids. Polynucleotides are made up of nucleotides, while lipids are made up of fatty acids and glycerol. Fatty acids may be saturated fatty acids or unsaturated fatty acids. Secondary metabolites may include alkanoids, phenolics, terpenoids, essential oils, toxins, and lactans. Inorganic material include prime elements, macro elements, trace elements. Prime elements we know are carbon, hydrogen, oxygen, and these are like water is also present. Macro elements include nitrogen, phosphorus, potassium, calcium, magnesium, and sulfur. Trace elements which are present in very minute amounts are boron, chlorine, zinc, copper, iron, sodium, molybdenum, nickel, silicon, and cobalt. So with this was about the theoretical part of today's session, and now we have few questions to answer. For example, first question is, which of the following is the most abundant element that is present in the human body? So options are carbon, hydrogen, oxygen, and nitrogen. So we studied that the most abundant element which is present inside the human body and in the earth crust as well is the oxygen. So the correct answer to this question is option number 3, oxygen. Next question is, what are anthocyanins? So we know that anthocyanins are the secondary metabolite products of plants, and these anthocyanins are so what are the options? First we will look at the options. So options are pigment or alkaloids, drug, or toxins. So we know that anthocyanins are nothing but the pigments which are produced by the plant cells. So anthocyanins are the secondary metabolites of the plant cells in the form of a pigment. So question number 3 is biomolecules are organic compound, inorganic compounds, volatile compounds, both 2 and 3. So we know that the biomolecules are the organic compounds which are present inside the living organism, and they are the non-living organic compounds that have a living function. The acid which was used for separation of biomolecules in a living tissue is, options are trichlorobenzoic acid, benzoic acid, trichloroacetic acid, and option 4 acetic acid. So we know that the process of extraction of biomolecules from the living tissue is to mix the living tissue with the acid, which is known as the trichloroacetic acid. Upon mixing, it will form a thick slurry. Then we will pass this slurry through the cheesecloth, and whatever gets filtered is known as the filtrate or the acid soluble pool. Whatever remain above the cheesecloth is known as the acid insoluble pool. So the correct answer is to this question is the trichloroacetic acid. So with this, I would like to end today's session and would like to thank you all for attending this. Thank you.